Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Ever think about why Alzheimer's disease kind of picks on some parts of the brain more than others, especially in the early stages?
Yeah, it's a really interesting question.
You've got all these different areas of the brain, right? With all these neurons all connected together, this massive network. And yet some of these areas are just way more vulnerable to those first signs of Alzheimer's damage, while others seem to be, protected, at least for a while.
It's almost as if Alzheimer's has I don't know, preferences for certain parts of the brain.
Exactly. And what's even more wild is that those super early signs that something might be off in the brain, like little changes in your sleep or maybe feeling more anxious than usual, those can actually show up way before any memory problems become noticeable.
Like decades before.
Decades. Yeah, exactly. It really makes you wonder, what's happening under the surface that we just don't fully understand yet.
It's a real mystery and a really important one to solve. Because if we can figure out what makes some brain areas so vulnerable early on, it could be the key to developing new treatments that could really change the course of the disease.
Absolutely. And that's exactly what we're going to dig into today. We're taking a deep dive into some really cool new research out of UC San Francisco. This study actually gets down to the molecular level to try and understand why some brain areas get hit harder by Alzheimer's than others right from the very beginning. They did this by looking at actual samples of brain tissue from people who had passed away and comparing two regions that are known to have very different vulnerabilities to Alzheimer's.
And this direct comparison within the same person is a really clever approach because it lets them control for all those individual differences that could be playing a role. Things like genetics, age, lifestyle. So it gives them a much clearer picture of what's truly different between those vulnerable regions and the ones that are more resistant.
Right. So today our mission is to really break down this study. What did they find? What are the key differences between those vulnerable and those resilient areas of the brain in those super early stages of Alzheimer's? And what could this tell us about how we might be able to treat this disease in the future?
Totally.
We're pulling our information from a paper published in Alzheimer's and Dementia, as well as a summary of the key findings from the UCSF Health News website. And we're going to try to connect all of this to, your interest in understanding this really complex disease.
It's a fascinating area.
So let's jump right in. What was the big question the researchers were trying to answer with this study?
Well, their main goal was to figure out, that basic level. Why are some groups of neurons so much more susceptible to those early changes that happen in Alzheimer's disease, while others seem to be, pretty unaffected, at least at first?
So they were looking beyond just the presence of, the amyloid plaques and tau tangles. They wanted to understand what makes some neurons more likely to be affected by these things in the first place.
Exactly. They were looking for those underlying mechanisms, the why behind the vulnerability. And to do this, they really dove into the details.
Okay, so how did they actually go about doing this? What was their methodology?
So they started with something really valuable.
Mm-hmm.
Brain tissue samples donated from people who had passed away. These samples came from two well-respected brain banks, one at UCSF and the other at the University of São Paulo in Brazil. And what's really cool about their approach is that they were able to compare two different brain regions within the same individual.
Oh, so they weren't just comparing brains from different people. They were actually looking within the same brain.
Precisely. And that's crucial because it really eliminates a lot of the variability that you might see between individuals. It allows them to zero in on those differences that are specific to those brain regions.
And which two brain regions did they choose to focus on?
They picked two regions that are located below the cortex, which is the outer layer of the brain. And these regions have some similarities, but they're known to have very different vulnerabilities to Alzheimer's. The first one was the Substantia nigra.
Okay.
Now, this region is responsible for producing dopamine.
Ah, dopamine, the feel-good chemical.
Exactly. And it plays a role in lots of important functions like movement, motivation, reward. And the interesting thing is that the Substantia nigra tends to be pretty resistant to the kind of cell death that you see in the early stages of Alzheimer's. So it's kind of like a tough cookie.
Right. So that was their more resilient region. What was the other one?
The other region they looked at was the Locus Coeruleus.
Okay.
And this area produces norepinephrine, which is involved in things like attention, the sleep-wake cycle, and our stress response.
Interesting.
Now, unlike the Substantia nigra, the Locus Coeruleus is actually highly vulnerable in early Alzheimer's. and it often shows signs of the disease even before areas like the hippocampus, which is involved in memory.
Wow, so it's like one of the first areas to be hit.
Pretty much. So they had these tissue samples from these two regions, one that's super vulnerable, one that's more resilient, and all from the same individuals. Now, what they did next was really dive into the molecular level to see what was going on.
Okay, get ready to get technical on us.
All right, here we go. So they isolated RNA from both of those brain regions.
RNA.
Yeah, RNA.
Okay.
And just to remind everyone, RNA is basically a messenger molecule that carries instructions from our DNA to the protein-making machinery in our cells.
Ah, okay.
So by measuring the levels of different RNA molecules, they could see which genes were being expressed more or less in each of these brain regions. And this gave them a snapshot of what cellular processes were happening.
So it's like looking at the activity log of the cell. You can see what's been turned up, what's been turned down.
Exactly. And what they were really looking for were differences in gene expression between the vulnerable Locus coeruleus and the more resilient Substantia nigra. Anything that could explain why they're so different.
And did they just stop at the RNA level, or did they go further?
They did go a step further to confirm their findings. They used a technique called immunohistochemistry.
Say that 5 times fast.
Right. So basically, this involves treating the brain tissue samples with special antibodies that bind to specific proteins.
Yeah.
And by visualizing where these proteins are located and how much of them there is, they could confirm whether those differences they saw in RNA levels actually led to corresponding differences in protein levels within the neurons.
Okay, so it's like double-checking their work. They saw differences in the RNA, which is like the blueprint, but then they wanted to see if those differences actually translated into different amounts of protein, which are the actual workers in the cell.
Precisely. So they did all this work. They compared the RNA, they looked at the proteins, and they found some pretty striking differences between the Locus coeruleus and the Substantia nigra.
So spill the beans. What did they find? What stood out?
Okay, so one of the most interesting findings was a huge difference in how these two regions handle cholesterol. Now, cholesterol is super important for brain function.
Wait, cholesterol? I thought that was a bad thing.
Well, in the context of heart health, too much cholesterol can be a problem. But in the brain, cholesterol is actually essential for things like building cell membranes, especially in neurons. And for processes like forming synapses, which are the connections between neurons, the brain actually produces its own cholesterol. And the way the cholesterol is transported and regulated within the brain is really complex.
So what was different about cholesterol in these two brain regions?
Well, they found that the Locus coeruleus, the vulnerable one, had gene expression patterns that suggested it had this really high demand for cholesterol. The neurons in this region seem to be actively making cholesterol and also taking it in from their surroundings.
So they're like cholesterol hogs.
You could say that, yeah. They seem to have this, insatiable appetite for cholesterol.
Wow. What about the Substantia nigra?
Well, the Substantia nigra, the more resilient one, didn't show that same level of activity related to cholesterol production and uptake. It's like they didn't have that same intense craving for cholesterol that the Locus coeruleus did.
That's wild. So one area is all about cholesterol, the other one's not so much. And this was reflected in the gene expression data.
Exactly. And it was also confirmed by the protein staining. They found higher levels of a protein called the low-density lipoprotein receptor, or LDLR, in the neurons of the Locus coeruleus.
Okay, what does that do?
Well, this receptor is part of a larger structure called the sigma-2 receptor, and it plays a key role in helping cells take in cholesterol from their surroundings. So the Locus coeruleus not only had genes that were suggesting high cholesterol production, but it also had more of the machinery to actively pull cholesterol in from the outside.
So it's like they were really set up to get as much cholesterol as they could.
Yeah, that's a good way to put it. And this is where it gets really interesting in terms of Alzheimer's disease.
Oh, here we go. Lay it on me.
Okay, so the researchers think that those toxic amyloid-beta oligomers, those clumps of amyloid-beta protein that are a hallmark of Alzheimer's,
Right.
they might actually be using this very same sigma-2 receptor with its LDLR component to get inside the vulnerable neurons of the Locus coeruleus. Because those neurons have more of these receptors on their surface, they might unintentionally be providing more entry points for the amyloid-beta to get in and cause damage.
Oh, that's sneaky, like a Trojan horse situation.
Exactly. Like having more doors to your house means there are more ways for those unwanted guests to get in.
And in this case, the unwanted guest is amyloid-beta.
Right. And what's really intriguing is that the Substantia nigra, the resilient region, It actually expresses a specific molecule that breaks down that LDLR.
So it's like it has a way of getting rid of those doors.
Exactly. And by reducing the number of these receptors on its surface, it might be less likely to let in those amyloid-beta oligomers, even if they're floating around.
That's amazing. So it's like this really delicate balance. One region needs a lot of cholesterol, but it might come at a cost. The other region has a way of limiting its cholesterol uptake. And maybe that helps protect it.
Yeah, it's really cool, right? And they didn't just look at cholesterol. They also did an analysis to look at broader patterns of gene activity across different biological processes. And this revealed even more differences between the two regions.
Like what?
Well, they found differences in areas like antioxidant defense mechanisms, a cellular communication pathway called KRAS signaling, and estrogen signaling.
So these two brain regions are like living in completely different worlds, huh?
It really seems that way.
Yeah.
And these differences could all be contributing to why they have such different vulnerabilities to Alzheimer's disease.
So this study really paints a picture of two brain regions that might seem similar at first glance, but they're actually operating in very different ways, especially when it comes to cholesterol. This is some pretty big stuff, right? What does all this mean for our understanding of Alzheimer's?
It has some pretty important implications. First of all, it really highlights how even very small differences in these fundamental cellular processes, like, how cells regulate cholesterol, can have a huge impact on why some people might be more susceptible to Alzheimer's disease than others. And importantly, why specific brain regions are affected earlier in the disease.
So it's not just about the presence of amyloid beta, it's about how individual neurons are interacting with it. And those interactions can vary a lot from region to region.
Absolutely. And this opens up a whole new avenue for thinking about how we might treat this disease.
You mean by targeting those specific interactions?
Yeah. I mean, imagine if we could create a treatment that specifically adjusts how cholesterol is taken up in the locus coeruleus, or maybe blocks the interaction between amyloid-beta and that LDLR.
So it's about finding those vulnerabilities and either protecting against them or exploiting them to stop the disease in its tracks.
Precisely. It could be much more targeted and potentially more effective than our current strategies.
That's super exciting. And the study also mentioned that the vulnerability of the locus coeruleus might be linked to things like sleep problems and inflammation.
Oh yeah, definitely. The locus coeruleus is a major player in regulating sleep-wake cycles. And it also helps control neuroinflammation.
Which is basically the brain's immune response.
Right. And we're learning more and more that both sleep disturbances and chronic inflammation are significant risk factors for Alzheimer's disease. And they can actually speed up the disease's progression. So if we can find ways to protect the locus coeruleus early on, well, it could have these ripple effects that benefit other aspects of brain health as well.
That's a really holistic way of thinking about treatment. You're not just targeting the amyloid beta, you're also trying to preserve those critical brain functions that are affected early on.
Totally. It's about taking a multi-pronged approach. And the fact that amyloid beta might be using that cholesterol uptake system as a way to sneak into vulnerable neurons, that's a huge clue for drug developers.
Yeah, it's like they've found a specific back door that the amyloid beta is using to get in.
Exactly.
Yeah.
And if you can figure out how to lock that door, we might be able to keep those harmful proteins out.
Now that's some seriously cool science, but it's important to remember that no study is perfect, right?
Yeah.
Did the researchers mention any limitations to their work?
Oh, for sure. Like with any research, there are things to keep in mind. For one, they were using postmortem brain tissue.
Meaning tissue from people who had already passed away.
Right. And while that kind of tissue is incredibly valuable for understanding what's going on in the human brain, it's just a snapshot in time.
Yeah.
And there could be all sorts of factors that influence the state of that tissue after death, like the person's condition before they passed away or how long it took to process the tissue. The researchers did their best to account for these factors by using matched samples within individuals and making sure the RNA they were analyzing was high quality.
Okay, so they were aware of those limitations and tried to control for them as much as possible. What else?
Well, the way they analyzed the RNA was at a bulk level, meaning they were looking at the average gene expression across all the different cell types in the tissue sample. Not just the neurons, but also the glial cells, which are the support cells in the brain.
Right.
So while they were careful about drawing conclusions about processes that might be really influenced by differences in cell type proportions, like inflammation, they did find some evidence that there were some variations in the proportions of different cell types between the locus coeruleus and the Substantia nigra. So Future studies that look at gene expression in individual cells.
Single cell analysis.
Exactly. Could provide even more detail and nuance to the findings.
Okay, so that's something to keep in mind for future research. Were there any other limitations?
Well, one last thing is that the study identified potential pathways and connections. But we have to remember that correlation doesn't always equal causation. Just because two things are happening together doesn't necessarily mean one is causing the other. So while the findings strongly suggest a link between cholesterol processing, the levels of that LDLR, and the vulnerability of the locus coeruleus, more research is needed to prove that those factors are actually directly causing that region to degenerate in Alzheimer's disease.
It makes sense. It's about building that evidence base. Did the researchers have any thoughts about that or any key takeaways from their work?
Yeah, for sure. So Dr. Alexander Ehrenberg, the first author of the study, he emphasized how remarkably similar the locus coeruleus and the Substantia nigra are, despite their very different vulnerabilities.
So even though they're both affected by Alzheimer's in different ways, they're actually quite alike in many other ways.
Exactly. And he pointed out that this suggests that even very subtle differences in their baseline state, like, for example, how much they rely on cholesterol, could be absolutely crucial for determining how susceptible they are to the disease.
So it's these tiny little initial differences that could end up having these big consequences down the line.
Yeah, it's a powerful idea.
And what about Dr. Leah Grinberg, the senior author? What were her thoughts?
Dr. Grinberg emphasized the broader implications of the findings, suggesting that the way the brain handles cholesterol might explain not only why some people are more susceptible to Alzheimer's than others, but also why specific brain regions are hit earlier.
So cholesterol could be playing a much bigger role in Alzheimer's than we previously thought.
Right. And she also stressed that understanding why the locus coeruleus degenerates and finding ways to protect it could have a huge impact on how we treat Alzheimer's disease, given its role in sleep and inflammation and all these other critical functions.
Protecting that vulnerable area could really change the game.
Totally. And, they also suggested that the processes they identified, especially those involving cholesterol and the LDLR, those could be potential targets for new therapies.
So it's not just about understanding the disease better, it's also about using that knowledge to develop new treatments.
Exactly. And it's worth noting that in their paper, they highlighted the importance of their study because it focuses on these subcortical neuromodulatory structures, which are these regions beneath the cortex.
Like the locus coeruleus and the substantia nigra.
Exactly. These areas haven't been studied as much as some other parts of the brain in the context of Alzheimer's disease. So their work is really shedding light on these crucial, early affected regions.
And as we wrap up our deep dive, what's the one thing you really want our listeners to take away from all this?
I think the biggest takeaway is this idea that even these seemingly basic cellular processes, like how our neurons handle cholesterol, can have these really profound and selective effects on brain health and how diseases like Alzheimer's develop.
It's like this domino effect. These little changes at the molecular level can set off this cascade of events that can ultimately lead to something as devastating as Alzheimer's disease.
Absolutely. And it's a reminder that even before we see any obvious signs of cognitive decline, there are these subtle changes happening that could be setting the stage for what comes later.
It makes you wonder, doesn't it? Since Alzheimer's seems to target these brain regions that control things like sleep and mood early on, could we be looking at those early changes in those functions as potential indicators of the disease, even before memory loss becomes the big problem?
It's a really fascinating question, and it's one that researchers are actively investigating. This whole area of research is incredibly exciting because it's opening up these brand new ways of thinking about how to detect, treat, and maybe even prevent Alzheimer's disease.
Well, that's definitely a hopeful note to end on. And I think this deep dive has really opened our eyes to the complexity of this disease and the incredible work that's being done to unravel its mysteries.
Absolutely. Yeah. It's a challenging disease, but the more we learn, the closer we get to finding effective solutions.
And that's what it's all about.
Couldn't agree more.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.